will be Jonathan Jeffet from Tel Aviv University, and he will tell us about spectral imaging. So whenever you want, Jonathan.
Okay, thank you very much. Can you hear me?
Yes.
Okay, great. Thank you for the organizer for this amazing online conference. I really enjoy it. I'm Jonathan from the Ebed State group in Tel Aviv University. And I want to show you in this talk our new system, CoCoS. It's a new method to see colors. So basically, as a microscopy community, you're all aware that color is a great contrast mechanism. You can distinguish between biological entities in complex environment and record their dynamics, like in this tracking of these membrane protein interacting on the cell. You can also use color spectroscopy in order to measure dynamic sub-nanometer distances and characterizing protein folding using FRET. So basically, you want to have as many color with the best temporal resolution. And to do that, you have three main schemes for color detection today. The basic and the most simple one is using a filter wheel, where you consecutively image different colors with different emission filters. Obviously, this gives you the widest field of view, but you pay with time, with your acquisition time, as each color is imaged consecutively. Another huge problem is that each color is imaged in a different time. Therefore, recording multicolor dynamics is problematic. The second approach, solving that, is to split your field of view between the different colors. With this method, you record all the colors simultaneously, but you lose throughput because of the real estate you take on your camera CCD. The most recent approach is to split the field of view into two spectral and spatial images, localization image and spectral image, by splitting your emission pathway into two and inserting a dispersive element in the spectral image. This solves the time issue and introduces much more color than the split view, but you lose, again, space, you pay with space, and also with SNR because you split the photons into two channels. So we wanted to solve all these problems and to reach to the optimal method, and obviously I'm presenting it here. So meet our CoCoS system. Basically, this is just above, it's just an epifluorescent microscopy with 5 laser excitation. This microscope image is inserted into the CoCoS add-on, which composed of a telescope and 2 prisms. And basically, the result of this CoCoS system is an image recording all colors simultaneously, encoding the color into spatial displacement along a single axis. So in this example, you can see 100 nanometer beads labeled with 5 different dyes. The dye spectra is over here at the bottom. And the filter bands that we used are in gray over here. So with this method, you get a really large field of view with almost no photon loss because these are refractive elements. You have simultaneous multicolor acquisition, and it's just a simple add-on to any microscope that maintains the optical axis and reduces the need for alignment. So let me explain a little bit more about how this works. The main building blocks are Amici prisms, these prisms that maintain a central wavelength that is passed through undiverted. Longer or shorter wavelength will be dispersed in opposite directions relative to this axis. But a single prism creates much too dispersion for our needs. So luckily, most optical problems that we're facing in bioimaging were once solved by astronomers. So they also solved this problem. If you introduce 2 prisms instead of 1, then you can, and you rotate one of the prism 180 degrees relative to the other, you can completely cancel all the dispersion. And if you tweak this angle, then you can continuously control the dispersion according to this equation. So basically, this gives you the opportunity to control your spectral resolution. So this is an example of a bead, a 100 nanometer bead labeled with 4 colors. And here I tweak the angle with motorized rotators of these prisms. So basically it allows me to have to move from fully canceled dispersion, a localization image, to a dispersed image where the spectral signature of each of these dyes is apparent. And also this shows how the compromise between SNR and throughput and resolution, spectral resolution, where you introduce higher spectral resolution, you lose SNR and you lose the throughput of your system. This continuous dispersion allows us to tweak between 3 modalities of CoCoS. You can do localization images without any dispersion. You can have color detection with just the minimal dispersion, allowing you to resolve the different colors that you use, or you can do spectroscopy with fully detailed spectra. You can also align the dispersion, the spectral resolution according to your experiment. So if you have 2 different dyes and you want to distinguish between them, you can calculate the exact relative prism angle to get you the perfect best SNR spectral resolution. So basically, let me show you that this works. Here are a few applications and examples for this system. The first application is the nanostring barcodes, showing color detection with the CoCoS system. Nanostrings are commercial molecular probes that are designed for RNA expression analysis. These probes are labeled with 4 different colors at 6 different positions, forming a unique barcode for each RNA molecule, target RNA. So with this commercial nanostring system, you can detect up to 800 different RNA samples in a single experiment. So here you can see how you see that on a commercial system. So you do sequential imaging switching filters, and you have your barcode detected. And this is how you see it in our CoCoS system with the optimal dispersion. So in a single snapshot, you can detect all colors and differentiate between the barcodes. And this gives you the ability with this matrix decoding imaging enables you to have a larger throughput because you just need quarter of the acquisition time to get all the colors. And also because you don't pay any more time for each additional color, you can introduce more colors and have up to 400-fold increase in available barcode combinations relative to the commercial system. So this is a big application for next generation. So the next application is using spectroscopy, and we wanted to see if we can resolve different very close by spectra of different dyes. So to do that, you need to do 2 sequential imaging, one with without dispersion, a localization image, and then to change the prism angle to get a spectral image, dispersed image. And if you take the intensity profile in this image and you compare it to the displacement from the localization image, you can deduce the entire full spectral information of each molecule. So to show this, I labeled 100 nanometer dyes, beads, with 3 far red dyes. These are the dyes, and mix them together on the same field of view. And here you can see an overlay of the localization in white and the spectral image in red. And here are 3 examples of 3 different beads or different dyes. So using cross-correlation comparison, I can classify different dyes according to their spectra. So this is the full classified field of view of the beads. And here you can see an overlay of the spectra of single beads showing that we can resolve 6 nanometer differences in spectra. And this is really expands the color palette that we can use for future applications. So with under 6 nanometer spectral resolution, you can distinguish 6 different dyes in the same spectral window that you usually use for Psi-5 molecules. This really reduces the optical complexity and reduces the photo damage because you only need 1 single laser excitation. The last application is measuring nanometer distances with single molecule FRET. So in FRET, when you excite a donor dye, labeled here in green, the acceptor dye can steal its emission photons and emit them in longer wavelengths. The efficiency of this process, this FRET process, is highly dependent on the distance between the different donor and acceptor dyes, allowing to measure sub-nanometer distances. In this experiment, we used synthetic DNA molecules tagged with specifically distanced donor and acceptor dyes, resulting in low, medium, and high FRET efficiencies. And here is how you see it in CoCoS. So the emission of the acceptor is dispersed above the emission of the donor. So you can see both emission simultaneously on the same field of view. And here is a time trace of the donor and emission of the donor and acceptor emissions with donor excitation, showing the different FRET efficiencies. Using alternating laser excitation, we can accurately quantify the FRET efficiency and resolve the different molecules in our sample. So with this, you can do single molecule epi-illumination, single molecule FRET, which is something you don't really do normally. And you can really increase the throughput of the standard dual color FRET and also do multiple color really easily. So all of this is now published in bioRxiv. I want to thank all our collaborators and especially Yuval, my awesome PI here in Tel Aviv University, and our great team, and also the funding from Azrieli Foundation giving me their generous fellowship. And that's it. I hope you have questions and I'll be happy to answer them.
So thank you very much, Jonathan. Let me start my video. Okay, so we are almost done with the session, but and I don't have any question in the chat, but I have one. What about overlapping? I mean, when you have two beads that are in the same direction that you are dispersing light, in that case, you have a kind of crosstalk. And what do you deal with that? Well, how do you deal with that?
Okay, so basically every method has caveats, right? So this is our caveat. The basic problem with our system is that you need to disperse your image so in one direction, but you can always change the direction of the dispersion according to what you need. So if you have some kind of, you need to be dispersed enough, like in Storm or in Palm, but you can change the direction of the dispersion as you will with just a press of a button.
Okay, so I think that we have to close here, but thank you very much with your for your presentation. I think that it has been very interesting to see your work. And okay, I just am a reminder for the rest of the people, and that okay, right now the Meet the Speaker with the previous invited speaker Robert Prevedel is open, the room. So please go there if you have some additional questions to the talk. And okay, thank you for coming to this session.